Please welcome to this video on lysosomal protein sorting and targeting. In this video, I plan to present one definition of lysosomes and the features of lysosomal enzymes, two requirement for the synthesis and modification of lysosomal enzymes in the rough endoplasmic reticulum, and three, a detailed mechanism of sorting and targeting of these enzymes. So what are lysosomes and lysosomal enzymes? Couple of key features about this aspect. Lysosomes are membrane-bound cell organelles with acidic luminal pH. They contain hydrolytic enzymes or hydrolases which function at acidic pH, and they hydrolyze or break down the macromolecules such as DNA, RNA, protein, lipids, and carbohydrate into their monomeric units in the acidic pH. Therefore, these all organelles play a significant role or key role in degrading damaged organelles, pathogens, and waste materials. I will present the overview of the lysosomal proteins and enzymes which start their synthesis on free ribosomes, how they reach the lysosomes finally. The first step is integration of free ribosomes engaged in the synthesis of lysosomal enzymes and protein with the rough endoplasmic reticulum for their synthesis and modification. In the second step, these modified enzymes and proteins are transferred to Golgi apparatus for further modification, and they receive in the cis Golgi sac an important or key marker which is Mannose 6-phosphate. So Mannose 6-phosphate is a key marker of lysosomal enzymes, and finally they reach trans-Golgi network from where they are sent to the early endosomes in the form of clathrin coated vesicles. These endosomes matures into late endosome which finally deliver the content into lysosomes. So I will discuss the mechanism of their synthesis, modification, and targeting to lysosomes. Here I present a very brief overview of how the free ribosomes engaged in the synthesis of lysosomal enzymes integrate into the rough endoplasmic reticulum. They integrate by utilizing the signal peptide present at the end terminus of all the lysosomal enzymes and proteins. However, for a detailed description on this aspect, I will advise you to watch our dedicated video on our YouTube channel called Future Academics. Based on the presence or absence of Mannose 6-phosphate marker on the lysosomal proteins or enzyme, they can be divided into two categories. The first one include lysosomal hydrolases. This is the major class. These enzymes or proteins, they carry Mannose 6-phosphate as a marker which is added in the cis Golgi sac. This is the major class, as I mentioned, of lysosomal protein, and the second one includes those proteins enzymes which do not carry Mannose 6-phosphate tag. This includes beta-glucocerebrosidase, prosaposin, GM2 activator protein, and lysosomal integral membrane protein or LIMP2. So lysosomal proteins are sorted out in the trans-Golgi apparatus using three mechanisms or three pathways based on the presence or absence of Mannose 6-phosphate tag on these proteins or enzymes. The first one is MPR dependent or Mannose 6-phosphate receptor dependent. This is specifically designed to bind with the Mannose 6-phosphate tag on these proteins, and this is for the soluble hydrolases or other proteins, lysosomal proteins. So this is the major pathway for sorting lysosomal proteins. The second one is designed for beta-glucocerebrosidase, which do not carry Mannose 6-phosphate tag, and this is mediated by lysosomal integral membrane protein type 2 or LIMP2 dependent pathway. The third one is mediated by sortilin dependent pathway. This is for GM2 activator proteins, prosaposin and acid sphingomyelinase. These proteins also do not carry Mannose 6-phosphate marker. Therefore, you can call second and third pathway as MPR independent pathways. So I will discuss these pathways individually in detail. So the first one is MPR dependent pathway leading to the targeting of lysosomal proteins. So this start in the trans-Golgi network here. This has lysosomal enzymes, hydrolases carrying mannose 6-phosphate, and they are sorted with the help of mannose 6-phosphate receptor or MPR here. These proteins they bind to the receptor, induces clathrin coated vesicle formation, which uncoat and fuse with early endosome. Early endosomes have internal acidic pH which disrupt the receptor from the enzyme hydrolases, and this early endosome matures into late endosomes which ultimately transferred or deliver the protein to lysosome. The receptor here is recycled back to the trans-Golgi apparatus, trans-Golgi network using retromer and TIP47 coactivator protein. So this is the mechanism how hydrolases carrying mannose 6-phosphate are delivered to lysosome finally. So let us discuss the LIMP2 pathway mechanism for beta-glucocerebrosidase. It starts in the trans-Golgi network also. You have enzyme here, beta-glucocerebrosidase, which do not carry mannose 6-phosphate. There is a receptor called LIMP2 with which these enzyme will bind and induces the formation of clathrin coated vesicles which uncoat and fuse with the early endosome. Early endosome has pH acidic pH which dissociate enzyme from the receptor, and this mature into late endosome which finally deliver these protein into the lysosome. So this is the mechanism how a protein or enzyme such as beta glucosidase without mannose 6-phosphate marker is delivered finally to the lysosome. The other point to note here is that the receptor here is not recycled, it becomes part of the lysosomal membrane. So let us discuss the third pathway, which is sortilin dependent pathway or sortilin mediated pathway. This is designed for prosaposin GM2 activator protein or acid sphingomyelinase enzyme. So this also starts in the trans-Golgi network. Here you have enzyme or these proteins are here. So these proteins basically are selected with the protein membrane bound protein called sortilin, and this also binding of these enzyme with the sortilin induces clathrin coated vesicles which uncoats and fuse with the early endosome which has pH acidic pH which liberates the enzyme from the receptor. These early endosomes mature into late endosome which deliver the protein ultimately into the lysosome. The receptor here is also recycled using same vesicles, clathrin coated vesicles. So there is a recycling also using retromer. So this is the third mechanism, and this is also a minor or alternative pathway for targeting lysosomal proteins. Clinically, the topic is very important because a defect in the targeting of lysosomal protein or enzymes causes diseases which are known as lysosomal storage diseases. Consequently, lysosomes accumulate the biological wastes leading to their enlargement and disruption of other cellular organelle function. These disorders are collectively known as lysosomal storage disease. Eye cell disease or mucolipidosis and Gaucher disease are lysosomal storage disorders characterized by defective lysosomal enzyme activity. resulting in substrate accumulation and associated clinical symptoms. All organs and tissues are affected in these conditions. In eye cell disease, particularly, the enzyme lack mannose 6 phosphate. As a result, the enzymes are mistargeted and therefore causing eye cell disease. Whereas in Gaucher disease, the enzyme beta glucocerebrosidase is defective or mutated and it is non-functional, leading to the symptoms of Gaucher disease. So let me pose a question and see if you can answer that. The question is, what type of glycosylation is mannose 6-phosphate, which is present on the lysosomal enzymes? The answer is that this is an linked glycosylation which starts in the rough endoplasmic reticulum and finalized in the cis Golgi sac of Golgi bodies. Thank you very much for watching this video. Please like it, share it and subscribe it. Until next time, you take care. Bye bye.